Now's the time for a great deal on a new Honda. It's time to drive smarter with high-tech features and excellent reliability. Check out the Honda Civic, Accord, or HR-V. For a limited time, well-qualified buyers can get a 4.99% APR on a 2025 Civic or a 3.99% APR on a 2025 Accord or HR-V.
See dealer for financing details. Exclude Civic Si and Type R.
Have you ever had to put your plans on hold due to symptoms of generalized myasthenia gravis, or GMG? Like taking that weekend trip, talking with friends, or enjoying a meal. Learn about a treatment option that may help. Visit treatgmg.com to learn more. That's treatgmg.com.
Biologist Joe Hin Chio stormed into his boss's office one day in 1956, waving a copy of a paper they had recently written. It certainly was not the worst day of Chio's life. He'd spent 3 years in a concentration camp being tortured during World War II. It's hard to get worse than that. But Chio was still furious. The paper concerned human chromosomes. Chio thrust the paper in his boss's face, shaking with rage. "Why are you first author and not me?" he demanded. His boss, Albert Levin, tried explaining, but Chio would not back down, and things escalated quickly. Chio threatened that if Levin didn't make him first author, he would destroy their lab's research. He would trash all of the cells they used, all the slides, everything. Levin sat there stunned. Was Chio serious? He was. Chio was tired of being pushed around. And if he couldn't be first author on this paper, he was absolutely willing to go nuclear. From the Science History Institute, this is Sam Keene and the Disappearing Spoon, a topsy-turvy, science-y history podcast, where footnotes become the real story. To understand what made Joe Chio so angry, we have to go back to the work of another, older biologist named Theophilus Painter. Painter lived and worked in Texas. At the lab bench, he was known as a tinkerer. He once said that every good scientist should be able to take apart a Model T and repair it with nothing but pliers and some wire. In the early 1920s, Painter determined that mammals use the X and Y chromosomes to determine sex, a major discovery. But he's primarily known today for a highly embarrassing mistake. In 1923, Painter began investigating a deceptively simple question. How many chromosomes do human beings have? Chromosomes are bundles of DNA in our cells. All animals have a different number, and that number can vary widely. Some ants have 1 chromosome. Koalas have 16. Cats have 38. Dogs 78. The Atlas blue butterfly has around 450. There's a fern with 1,440 chromosomes. But for decades, biologists did not know how many chromosomes human beings had. Guesses ranged from 8 to 64. Now, you might be thinking, wait, how hard can this be? Just get a cell, find the chromosomes, and start counting. If only it were that easy. First of all, chromosomes are tiny, visible only under microscopes. They're also the same color as the rest of the cell. You have to dye them to make them stand out, and even then, they're still hard to see. Worst of all, chromosomes in cells are normally long and skinny. They're like spaghetti noodles, only millions of times longer. They squiggle and overlap and twist all around each other. It's hard to know where one stops and another begins. Imagine being handed a bowl of spaghetti and having to guess how many noodles were in it. Would you even get close? That's essentially what biologists faced when counting chromosomes. But there was one trick. As you might know, cells divide sometimes. You might remember pictures from high school biology. A cell copies its chromosomes, then all the chromosomes line up in the middle of the cell and get pulled opposite directions. The key point is that during this stage of division, chromosomes are far more compact. They cinch together. They tighten up. Not completely. There's still some spaghetti overlap, but it is easier to count than before. So that's what Theophilus Painter decided to do. Find human cells that were dividing, then inject wax into them to gum things up and freeze them. Then he could try counting chromosomes. The only problem was adult cells do not divide very often. They mostly just sit there. In fact, the only real exception to this rule is the cells in the testicles that produce sperm cells. Men produce a lot of sperm, millions per week. And the sperm precursor cells in the testes are constantly dividing like crazy. So if Painter wanted to catch cells mid-division, testicle cells were his best bet. But this only introduced other problems. Those testicle cells had to be fresh. After death, chromosomes clump together, which again makes it hard to count them. So Painter needed very fresh cells, harvested right after someone died. To compound the problem, people just didn't donate their bodies to science in the early 1900s. Dissection was considered taboo, which made securing human tissues nearly impossible. To get around this taboo, biologists did scandalous things. Some actually lurked near the gallows when criminals got hanged and harvested their testicles. Painter drew on another squeamish source. A former student of his worked at a local insane asylum. Doctors there sometimes castrated inmates, surgically removed their testicles. So Painter hit his former student up. In 1923, the student castrated 3 men. In each case, he quickly diced up the testes, then dumped them in preservative fluid within 30 seconds of their removal. Afterward, he rushed them to Painter's lab. Painter had no other way to get fresh material. Of those 3 castrated men, 2 were black, 1 was white. After injecting the cells with wax, Painter sliced them into thin microscope slides with a special knife that he had invented that consisted of stacked razor blades. Unfortunately, if you're trying to count something that's already hard to count, it is not a great idea to chop it up into pieces, as Painter discovered the hard way when he studied his slides. Things were still messy. Sometimes he counted 46 chromosomes, sometimes 48. He kept counting and recounting, making himself cross-eyed following the spaghetti strands, and he still could not decide. He finally took a deep breath and made an educated guess. He published a paper declaring that humans have 48 chromosomes. Biologists were happy to have the number resolved, even on what amounted to a coin flip. And Painter's paper did have one positive effect. It established that black people and white people have the same number of chromosomes. That refuted a nasty theory going around that black people had fewer and might therefore be a different species. Painter declared otherwise. After that, 48 became enshrined as the standard number for humans, and Painter's results began appearing in textbooks, and they would continue to appear year after year, decade after decade, until Joe Chio got involved. Chio was born on the island of Java in 1919. He lived under Dutch colonial rule there, and he could speak French, German, English, Dutch, and a native Javan language. His father worked as a photographer, and Joe grew up helping out in the darkroom. But he gravitated more towards science, and by the 1940s, he was working as a research biologist, focused on breeding disease-resistant potatoes. That research stopped abruptly in 1942, when Japan invaded Java during World War II. As someone of Chinese ethnicity, Chio was thrown into a concentration camp and tortured. Despite this brutal treatment, he spent his time comforting fellow inmates. He even knitted underwear and sweaters for them to replace their ragged clothes. An act of real decency in a ghastly place. After World War II ended, Chio quit Java and took a Red Cross relief ship to Holland. After recovering his strength, he resumed doing scientific research, bouncing around to labs in Copenhagen, Spain, and Sweden. It was in Sweden where he met Albert Levin at a university in Lund. He and Chio began collaborating, with Levin as the senior scientist and Chio the junior technician. Over the next decade, they published 5 papers, always with Levin as the first author. Levin had started his career on plants, breeding sugar beets and clover. By 1955, he was studying how certain chemicals damaged the roots of plants. Specifically, the chemicals damaged the root cells' chromosomes. And something about the damage struck Levin. It looked identical to the damage he had seen in the chromosomes of cancer cells. This intrigued Levin. So he decided to study the links between chromosome damage and cancer. He asked Chio to join him. Now, in 1955, the study of chromosomes had advanced a lot since Theophilus Painter's day. Biologists had new dyes that made chromosomes easier to see inside cells. Biologists had also invented a preparation technique called the squash. Instead of infusing cells with wax and slicing them, you smushed them under a slide. That kept chromosomes whole and intact. Levin had found a neat trick as well. The crocus plant produces a chemical called calcicene. Calcicene happened to arrest cells right when they started dividing. Just sprinkle a little in, and cells froze right when the chromosomes were the most compact and the most easy to see. Perhaps most important of all, Chio and Levin did not need to lurk near gallows to get human tissues. Sweden was a rare country back then where abortion was legal, and Levin had access to fetal tissue. That was important. Again, beyond sperm cells, adult cells in humans do not divide often, but fetal cells do. As the fetus grows, cells multiply like mad. So with fetal cells, Chio and Levin had lots of chances to catch cells dividing. Then they could freeze them with the calcicene. Now, it's not clear why Chio began counting chromosomes in the fetal cells. Perhaps just as a warm-up exercise. Make sure everything looked right before they moved on to the more important matters. But that's not how things worked out. The warm-up exercise became the prize, the most important aspect of the entire project, and it would destroy the two men's friendship in the process.
Now's the time for a great deal on a new Honda. It's time to drive smarter with high-tech features and excellent reliability. Check out the Honda Civic, Accord, or HR-V. For a limited time, well-qualified buyers can get a 4.99% APR on a 2025 Civic or a 3.99% APR on a 2025 Accord or HR-V.
See dealer for financing details. Exclude Civic Si and Type R.
Have you ever had to put your plans on hold due to symptoms of generalized myasthenia gravis, or GMG? Like taking that weekend trip, talking with friends, or enjoying a meal. Learn about a treatment option that may help. Visit treatgmg.com to learn more. That's treatgmg.com.
Joe Chio was a night owl, often working straight through until dawn. He also worked through holidays, so there was nothing unusual about him firing up his microscope at 2 a.m. a few days before Christmas in 1955. He began examining slides of fetal lung cells. After working in his father's darkroom as a child, he still loved photography, and he used a special camera to snap pictures of what he saw through the scope. Then he started the warm-up counts. How many chromosomes were there? To his confusion, he found just 46. That puzzled him, so he grabbed a photograph of a different slide and counted that one. 43, 44, 45, and got 46 again. So he grabbed another photo and another. He eventually counted over 250 slides, by the end of which he was certain. Theophilus Painter and all the textbooks were wrong. Humans did not have 48 chromosomes. They have 46. Just try to imagine the thrill here. Chio had had a solid but unspectacular career before this, working under Levin's shadow. Now, at age 36, he had discovered something epical. It's what scientists call a golden moment, that heart-pounding instant when they know something that no one else in history ever has. And Chio surely would have rushed to tell Levin this, except Levin was on vacation at the time, nowhere near the lab. Chio waited impatiently for the holiday break to end and then told him. Levin was thrilled as well, and they agreed to rush something into print. For whatever reason, Levin took the lead writing the paper, and when he showed a draft to Chio, Chio was shocked to find that Levin had listed himself as first author. Now, why was this such a big deal? Because credit for discoveries means prestige for scientists. It's their currency. Scientists often work long hours for lousy pay. Credit for discoveries is the one thing they have. And here, Levin was trying to usurp credit for Chio's discovery. When Chio asked, "What the hell?" Levin got patronizing. He reminded him that that's just the way things were done. Senior scientists always got top billing, as Levin had on their previous 5 papers. But this 6th time, Chio put his foot down. He'd been toiling away at 2 a.m. all by himself, while Levin was off drinking eggnog and singing Christmas carols. Chio snapped, and he told Levin that if he wanted credit, he should have been in the damn lab himself. Annoyed, Levin pushed back. He asserted his seniority again. So Chio decided to go scorched earth. He threatened to destroy all the slides, all the cells, everything, if he did not get his name first. Talk about a tantrum. But however indignant, Levin backed down. He let Chio switch their names and thereby secure his place in history. But Levin's capitulation could not save their relationship. Chio simply couldn't trust Levin anymore. And Levin resented Chio, especially after their paper's publication made Chio a scientific star. So Chio skipped out to the United States, which had less hierarchy in its labs. Chio ended up working 37 years at the NIH in Maryland, mostly on cancer and genetics. Along the way, President John F. Kennedy gave him a medal for his work. Looking back on Chio's discovery, it's kind of shocking how long it took. Remember, it happened in 1956. Scientists by then had already discovered the famous double helix of DNA. But we still couldn't do something as simple as count the number of chromosomes we have. If Chio had not made the correct count, biologists could conceivably have gotten all the way to the Human Genome Project in 2001 without knowing. And if that sounds far-fetched, remember, authority casts a very strong spell on humans. It blinds us to what we can see. Chio actually went back later and looked in old textbooks at pictures of human chromosomes. And with his eyes opened, he could clearly see just 46. But people before him could not see what was right in front of them. For his part, Theophilus Painter was mortified to know that he had overcounted. The human chromosome count was supposed to be the seminal discovery of his life, what he was known for. And it was completely wrong. Talk about embarrassing. Chio's work on human chromosome number eventually opened up several avenues of research. One involved cancer, since again, cancer cells often have abnormal chromosomes. Another involved human evolution. Even though humans have 46 chromosomes, our closest ape relatives all have 48 chromosomes. Chimpanzees, gorillas, orangutans. It's a fundamental biological difference between humans and other apes. And if you're wondering how that's possible, how you just lose 2 chromosomes, I've actually put together a bonus episode at patreon.com/disappearingspoon. It's a fascinating glimpse into the emergence of humankind. The bonus also details a bizarre case from China of a man who has only 44 chromosomes, 2 fewer than most people. And the kicker is, he is perfectly healthy, a medical marvel. All that and more at patreon.com/disappearingspoon. Chio's work on human chromosome number also opened up research into genetic disorders that involve people having extra chromosomes. In fact, just 3 years after Chio's discovery, scientists in France built on his work to prove that Down syndrome results from an extra copy of chromosome 21. Unfortunately, that discovery also involved a senior scientist allegedly stealing credit from an underling. And this dispute got even more heated than Chio and Levin's. Even a half-century after the initial work, it was still raging, to the point that the underling's opponent sent law officers storming into a scientific meeting where she was speaking, using bullying and harassment to shut the meeting down and silence her. These opponents were essentially using the law to censor science, something that smacks of the Inquisition. And indeed, the man behind all this is now in line to become an official Catholic saint. It is a wild story, and we'll hear all about it in next week's episode. This is the Disappearing Spoon podcast, brought to you by the Science History Institute. Be sure to check out their in-depth science history podcast, Distillations. You can find it anywhere you get your podcasts or on their website, distillations.org. You can find more incredible stories in my books or at samkeen.com. You can also book me as a speaker there for your school or event. If you like this podcast, please spread the word and support it at patreon.com/disappearingspoon. You can also get bonus episodes and signed books at Patreon. This episode was written by me, Sam Keen. It was produced by Mariel Carr, Rigoberto Hernandez, and Sarah Kaplan. Our sound design, scoring, and mixing is by Justin Berger of Row Home Productions. Row Home's creative director is Alex Lewis. John Myers is Row Home's executive producer. Thanks for listening.
Row Home Productions.
Have you ever had to put your plans on hold due to symptoms of generalized myasthenia gravis, or GMG? Like taking that weekend trip, talking with friends, or enjoying a meal. Learn about a treatment option that may help. Visit treatgmg.com to learn more. That's treatgmg.com.